Dr. Jaime Tartar is a professor of neuroscience at Nova Southeastern University. She had earned her PhD in behavioral neuroscience at the University of Florida, where her research focused on long-term changes that occur in neurobiologic pathways involved in stress responses and developing animal models of stress. She also has completed postdoctoral training at Harvard, where she studied neurologic consequences of sleep perturbations using in vitro EPS, electrophysiology recording techniques. She received training in sleep medicine at Harvard, and she was awarded an honorary degree here at NSU. That's pretty cool. Dr. Tartar is the co-founder and CEO of the Society for Sports Neuroscience, an academic organization for the field of sports neuroscience, which I have to recommend if you all have not been to her neuroscience annual meeting. It is so amazing. Every time I've gone, I've walked away inspired and with a whole new great idea to pursue. This last time, now I have new friends in Germany. Thank you very much for that. We're doing some studies together. Dr. Tartar's current research is focused on stress, sleep, and athletic performance. And with no further ado, take it away.
So I'm here to talk to you today about sleep, some of the work that we've done in my lab on sleep. As Dr. Klimas said, I also study stress and human performance. So I was trained to study rodent models. And since I've been at NSU for, oh my God, it's been 19 years, I've switched over to studying humans. So let's talk to you a little bit about that. And hopefully you see the next slide, right? Okay, so everything's up and running just fine. So as Dr. Klimas mentioned, I do run a nonprofit organization where we kind of focus on some of the recent work and recent interests I have in sports neuroscience. And so I just wanted to promote our conference, which is going to be right here at NSU next year. We're just now getting this organized. It's going to be February 21st and 22nd. So we're still recruiting speakers, panelist ideas. So if you have any ideas, please go ahead and shoot me an email. I'd love to chat about it. Okay, on to sleep. So as a quick refresher, I'm sure everybody kind of remembers a sort of hypnogram and what happens during the night when we fall asleep. As a sort of super duper quick and dirty refresher, first thing we do, right, we jump down, try to get a lot of that slow wave sleep in early in the night, and going from slow wave sleep to light sleep to REM sleep, and kind of going up and down through about 5 or 6 cycles a night. And that first part, getting a lot of that deep slow wave sleep, which is so important for lymphatic activity, memory consolidation. And in the second part of the night sleep, not getting so much slow wave sleep anymore. So here we're focused a little bit more on REM sleep. And so we know REM sleep is also important for emotion processing. So we're going to come back, remember that, we're going to come back to that later on in the presentation. So for most of my career in human and non-human research, I've really worked on just doing terrible things, I feel, to humans and non-human animals. So giving them stress, giving them exercise when they don't want it, and taking sleep away. So what do we know happens when you lose sleep? Well, nothing good, right? Only good things happen to us when we sleep. So sleep, we really want to think about it as the Swiss Army knife of health. So only good things are going to happen. Things are going to improve when we're sleeping at night, and everything's going to be affected with sleep loss: cognition, mood, immune functioning, which is important for your group, brain health, hormone regulation. So we're trying to look at all of these different aspects in humans. Despite the fact that we know that sleep is so important for neural processing, for emotional processing, for immune processing, humans do something very strange, and we restrict our sleep. It turns out we're the only animal that practices sleep restriction. If you look at nature and animals kind of in the wild, even if you look at hunter-gatherer populations, they're not restricting sleep. In fact, if you look at hunter-gatherer populations, they don't even have a word for insomnia. So this is really an industrialized human issue. So I don't have to go very far to know this. I go home, my puppy, he's so cute, any excuse to get him into a slideshow. He likes to sleep, and my little puppy is not worried about staying up late at night, restricting his sleep, thinking about how much snacks he's going to get and how many kitty cats he's going to see the next day. Okay, so what's going to happen if we take sleep away from people in the form of total sleep deprivation? So we can start with that. So one of the first studies that we did is just acute sleep deprivation. These are young, healthy adults. They're easy access to them because they're students at NSU. So with this study, we basically gave them these active watches, which do a pretty good job of recording their waking activities and sleeping activities. And we did some baseline testing on them. Then we did an overnight sleep deprivation. We went ahead and followed them, did an overnight sleep deprivation where we both had the actigraphy monitor on them and we observed them. And then we tested them again on some of these measures. And so what did we see after one night of sleep deprivation in otherwise very young, very healthy people? Well, we have a significant increase in C-reactive protein. We have a significant decrease in cortisol levels, a significant increase in IL-6. Surprisingly, unlike some other studies, we didn't see changes to leptin and ghrelin levels. We didn't see significant changes to IL-1B. What is interesting, I think, with cortisol, sometimes people might think, oh, it's good to have low cortisol. Not necessarily. Right? Cortisol does a lot of important things in our body. One of the important things for your group, right, is like reining in the immune system. And you want cortisol levels to be high in the morning. So having this morning decrease in cortisol level isn't helpful. Now we're going to look at their mood using the profile of mood states. And basically what we see is across every measure of the profile of mood states, we're having mood impairments. That shows up in the overall total mood disturbance score as well. And then if we look at their anxiety levels after one night of sleep deprivation, significantly increased anxiety as well. So the mood disturbances I thought were particularly interesting, especially because we know from other work that even though we see this kind of emotional instability, mood changes with sleep loss, people are pretty bad at self-monitoring in general, and they're worse at it when they're losing sleep. And we kind of observed that during the study. We observed anecdotally some changes in mood and irritability in the research participants, and recognizing that they weren't necessarily seeing these changes in themselves. So as a neuroscientist, what I was really interested in now is getting into the brain. What's happening at the neurological level that may be explaining some of these mood changes? So with this study, we were looking at a really nice measure of mood and attention to emotional information. This is called the late positive potential. It's one of the event-related potential measures. And so if you look at just sort of the bottom of the slide where we see these kind of squiggly lines, the red squiggly line is really the person's neural response to a negative emotional picture. And the green one is their response to a positive emotional picture in that late positive potential range. The black line is just a neutral image. And so this is what we expect to see. We expect to see this positive going potential in the ERP when the person sees an emotional picture or has some type of emotional information presented to them. So we use the International Affective Picture System for this, showing them things like cute things, some things that are a little bit scary for the negative pictures. So relative to baseline recording, when we take sleep away from them in the form of 24 hours of sleep deprivation, again, through visual observation as well as actigraphy verified, we saw something really interesting. And what we saw is compared to that baseline day, and this looks like the graph that I just showed you, that we see they're not having an emotional response to neutral images, but they are to the red negative images and to the positive images in blue. After sleep deprivation, what we see happen is that these ERPs collapse. And so now they're basically responding the same way, independent of picture category type. So if we show them in such a way that if we show them a picture of anything, they're responding to it as if it were emotional. So we're getting a late positive potential response to all picture categories, which means we show them something very scary, we get an emotional brain response. We show them something really cute, like a puppy dog, we get an emotional response, but we show them a coffee cup and we also get an emotional response. So, and this tells us that the brain is kind of responding to everything in an emotional way. The neural generators for this come through multiple neural generators, but the amygdala is one of the neural generators for the late positive potential. And so what makes that particularly interesting is at the same time, we were having them rate pictures behaviorally. And so what was interesting is they were able to correctly rate the pictures. So they knew behaviorally, oh, that's a pen, that's not emotional, oh, that's a scary tiger, that is emotional. So that was all fine. It was that sort of subcortical brain processing that showed what would ultimately, I think, be observed as emotional instability, where the brain is sort of doing two different things subcortically. responding to everything as if it were emotional, even though behaviorally they could tell us the difference between the emotional and non-emotional pictures. We did a control condition in this as well. We just wanted to make sure there was no change in this through test and retest. So there was no effect of just having them do the task twice. The other thing that we looked at is just in the evening. So does it matter as we get sleepy throughout the day? Do we see this type of change as well? And we saw something very similar. So in the morning, we get that nice brain response where we're not responding to emotional pictures. We are responding to emotional pictures with an LPP. But then in the evening, not as dramatic as sleep deprivation, but we are starting to see the same pattern where now these light positive potentials are starting to collapse on each other. So that means that we're probably a little bit more emotionally unstable in the evening than we are during the day. So also looking at some biomarkers of health. So this is a study that we're still looking at. We did see differential expression of some of these microRNAs that do associate with cancer risk. We're continuing to analyze these data right now. So I would put out like a be on the lookout for the follow-up for this study. And the other thing that we looked at with sleep deprivation is glutathione, 5-MC, and sort of 5-HMC, ATP production, and cortisol. And here we see that same thing that we saw before, decrease in cortisol after one night of sleep deprivation, looking at those morning measures. So that increase in 5-MC, potentially showing heightened DNA methylation, the decrease in 5-HMC, looking at sort of that reduction in dynamic DNA demethylation, and changes obviously in oxidation, ATP. Okay, so that's sleep deprivation. Most of us, I don't think most of, well, hopefully since we've been in college, most of us are not doing 24 hours of sleep deprivation. These studies are very hard. They're hard on me to do. They're hard on the participants. Most of us are doing something else. And what most of us, I think, are doing is sleep restriction, meaning that we're not getting enough sleep on average every night. So maybe it's like 6 hours on Tuesday, 4 hours on Wednesday, 6.5 hours on Thursday, and then Saturday we'll go ahead and sleep in. So what does that do? This chronic sleep restriction. So one of the things that we looked at here, just looking at cortisol and the IL-1 beta cytokine, it looks like people, and these are people who are naturally restricting their sleep. So again, we're following them with activity over a long period of time. And what we see is if you're sleep restricting, as far as these two measures go, but you're going to bed early, that seems to be better than if you're restricting your sleep and going to bed late. There's lots of reasons why going to bed late is for the circadian changes, circadian misalignment that might occur with doing that, may be a little bit worse off for your health. So with cortisol, at least, evening measures of cortisol are increased in that group that has chronic sleep restriction with a sleep delay, so they're going to bed later, as opposed to IL-1 beta, which is increased in both the group that has just chronic sleep restriction, but then also in people who have chronic sleep restriction with sleep delay. And no, a lot of times we think, oh, the job of cortisol is to remain in the immune system. It's not necessarily doing that here. So maybe in that group, we're also maybe seeing a little bit of glucocorticoid resistance as well. Okay, what about sleep quality? So lots of ways to measure sleep. How good is your sleep when you're getting it? And does that matter? Sleep quality. Obviously it matters because I asked the question. So it'd be weird if the next slide was like, oh, it doesn't matter. So it matters. Let's see how it matters. So here we're looking at attention measures. And we all know, right? Attention is one of the most expensive things your brain can do. Humans are not paying attention like 50% of the time. We run this default mode network. So we're already not good at it. You put the brain in a vulnerable state. Oh, now we're really bad at it, right? So we take sleep away from people or give them stress, and attention suffers. So we see that here. We see the target accuracy decreases on an attention task in poor sleepers. People have poor sleep quality. But then the other interesting thing that we see is that sort of affecting mood again. We all go through life, human brains go through life with this sort of innate negativity bias, right? We are more sensitive to emotionally negative valenced information relative to positively valenced information. Our brains are just on the lookout for negative information and they're highly sensitive to it. So it kept their ancestors alive. All the happy people didn't make it. And here we are, the product of all those people who were worried all the time. So we take sleep away, that's going to amplify in these people with poor sleep quality. So not take sleep away, people with poor sleep quality. So they're just more likely than people with good sleep quality to recognize, to be sensitive to the negative stimuli. And they're also more likely to have false alarms for negative information. And then what about sleep efficiency? So this is a really important measure for a lot of us, because this is that measure of, for the time that you're in bed, how much of that time are you actually sleeping? Like, how efficient are you at sleeping while you're, ostensibly trying to sleep? So with this study, this is a study that we looked at the gut microbiome. So this was the original study. We went ahead and just looked at males in the study because we were a little concerned about potential sex differences here. And so what we saw, if we looked at that measure of sleep efficiency, looking at both the Shannon index, which both of these are really good measures of gut microbiome diversity. Maybe the Shannon index is a little bit better at richness and evenness. Maybe the Simpson a little bit more evenness. Maybe the Shannon's a little bit better at rare species, but they're overall really nice measures of gut microbiome, sort of just general diversity. So we see that there is a very clear relationship between sleep efficiency. So the people who are not as efficient at sleeping do not have the same richness and diversity in their gut microbiome. We looked at specific measures in this study as well, like specific taxa of gut microbiome. What was interesting is that if we looked at one particular phylum, like some of the taxa, some would go up with sleep efficiency and some sleep measures, some would go down. So there didn't seem to be like a really clear pattern of any specific taxa or phylum that associated, but they all seem to be doing different things, which sort of makes sense when you look at sort of how they behave as part of the gut microbiome and the gut microbiome brain axis. So the other thing that we thought was pretty cool was interleukin 6. So when we looked at interleukin 6, he just seemed to associate with everything in kind of interesting ways. So interleukin 6 really interacted, we thought, with the main outcomes of the study, but really interacting with each node. So in that study, we had done some cognitive testing, some emotion testing, and it was really interacting with those nodes, with the gut microbiome in general, and then with multiple measures of sleep efficiency, and sleep time and awakening and time in bed. So all these measures that we get from actigraphy. When we think about sleep, and I wanted to make sure that I put this slide on here, just one of those points that we always want to make when we're talking about how people respond to anything behavioral or how people respond in general. We always just want to recognize individual differences because they do tend to matter. So not everybody is responding the same way every time. And so we did a couple of studies just sort of wanting to clarify that these individual differences do exist. So for example, if we look at people who are genetic night owls with that per3 polymorphism, it's a repeat polymorphism, either repeats 4 or 5 times. So when you carry, or you're homozygous for the 4 repeat polymorphism, that makes you a little bit more likely to be a night owl. So we consider the night owl polymorphism. So even if you come and behaviorally, being a night owl doesn't associate necessarily with good behavioral health outcomes, but it seems like when you look at this, it also matters not just whether or not you carry that polymorphism, but what are you doing behaviorally? Because sleep is in many ways a modifiable behavior. So if you're getting enough sleep, having that polymorphism doesn't seem to be detrimental to your mood, depression, and anxiety scores. But if we're getting 6 hours or less, so now you're going to bed late and you're probably having to get up in the morning for work, or getting up in the morning for whatever reason, but you're not getting a lot of sleep, that's when we tend to see that polymorphism matters for increasing trait anxiety, tension, depressive symptomatology on 2 different measures, the CESD and the POMS. The other thing that we see is even just carrying polymorphisms associated with dopamine production. So if we look at the monoamine oxidase A polymorphism, so we see people who have higher dopamine, which we can associate with wakefulness, or in this case, low monoamine oxidase activity, also have poorer sleep quality overall. So these genes that aren't necessarily these canonical clock genes like dopamine, and some of our polymorphisms within the clock genes do seem to predict individual differences. Okay, so in the last couple of years, I've moved professionally into an administrative physician. Some would say against my will. I wouldn't say that because this is being recorded. But I moved into this chair physician, and the first year I did this, my research kind of suffered a little bit, but in a way I think it was good because it gave me some time to sort of reflect a little bit on my research and some of the work we were doing with stress and some of the work with human performance. And I just sort of felt like I was tired of doing mean things to people. I was just getting kind of tired of giving people stress and making them run against their will and taking sleep away. So I thought, well, maybe I could just do nice things to people and get at these questions from another perspective. You could call it a midlife crisis. I don't know. I just tired of doing terrible things. So let's see what happens if we give people sleep, right? What might we be able to do with this? So we did. We tried. This study was really hard. It took a long time. It took 3 years to do this study. Because the screening, because we really wanted to get people who were only sleeping, who were chronic sleep restrictors. And then we also wanted to get people that weren't coming into the study with mood impairment. So no depression, no anxiety, no sleep disorders. So they went through a clinical interview, a 1-hour clinical interview. And so we just couldn't get a lot of people that met that criteria. But when we did, we were able to give them baseline sleep monitoring. Then we took their average sleep, verifying that they were chronic sleep restricting. And you can't force people to go to sleep, it turns out. So with sleep extension, what we do is we monitor their time in bed, and those actor watches do a pretty good job of that. And we ask them to spend 90 minutes extra in bed. And it turns out when you do that, when you have people spend an extra 90 minutes in bed, they'll sleep longer. So we do the sleep extension, which is a 90-minute time in bed extension. And then we post-test them for the study. So we were able to significantly verify that through actigraphy that they did spend more time in bed and they did increase their total sleep time. So what happens if we give people more sleep that weren't getting enough sleep? Well, it turns out it is super bueno. I am working on my bilingualism. Super bueno for mood. So this is great because we have a lot of people, and this study involved a lot of young people. So as you probably know, depression and anxiety rates are sort of an all-time high. A lot of young people are suffering from mood disorders. So this was really nice to see. So when we look at their time in bed during sleep extension, we did see a significant relationship with all kinds of mood measures. So emotional support. So this is using the NIH toolbox measures, I should mention. Perceived hostility is down. Anxiety, the somatic symptom, perceived somatic symptoms are down. Anger significantly reduced, and anger hostility measures as well. So lots of improvements in emotional measures with that 90 minutes of sleep extension. We just wrapped up this study, and we just did the gut microbiome analysis. So don't tell anybody because we haven't written this up yet. So we're hopefully going to do it soon. But I did want to share that because it's kind of like it's one of those moments. So we didn't see the same gut microbiome species or bile or tax or any of those that we saw with the other study. This one we saw Peptostreptococcus, which is a little bit maybe a little bit surprising. It was, anaerobic, gram-positive bacteria. But if you do dig in a little bit, you can see that ultimately it can reduce gut systemic inflammation, and it can improve intestinal barrier function. But probably why we're seeing this relationship with this one is because it promotes tryptophan metabolism. So it can increase serotonin, which we know is important for not just mood, but also circadian regulation, and melatonin. Not necessarily a molecule I really like, I have another study of melatonin. I'm not showing for the sake of time, but I love melatonin. It's such a wholesome molecule. It just doesn't have really good PR, I think, but it's very safe, very wholesome, but it won't put you to sleep, right? So can help a little bit with sleep initiation, but really more of a circadian regulator. So melatonin does, so when you're traveling across time zones, right, that's where melatonin shines. But, you know, it's antioxidant, anti-cancer, anti-tumor. It's been around for 3 billion years. So potentially, I'm digressing, but potentially that's where that relationship might be. So potentially improved sleep environment with that sleep extension that we're doing, it might favor some of these beneficial microbiomes, microbes. So keep in mind, we're still looking at the data. So stay tuned. Hopefully we'll get this out by the end of the summer. So the other thing that I've become recently interested in the vein of doing nice things for people is non-sleep. Oh, that says non-sleep. Sorry about that. Hopefully we can edit that in the recording. So non-sleep deep rest. So non-sleep deep rest. There's some research on this, and this is the idea that if you can't sleep during the day, if you can't nap, could deep rest give you some positive outcomes? So this would be things like progressive muscle relaxation, meditation, and yoga nidra, which is sort of a sleep yoga process. There is other evidence to suggest that non-sleep deep rest can also promote lymphatic activation during the day. So we were really interested in this as well. So we just wrapped up this study earlier this year. So with this one, we had a professional yoga nidra, which is also called iRest, coach, and she was able to record for us a 30-minute yoga nidra session. So we had a smaller sample size, and this was just an initial associational correlational study. But what we did find is that the people, so we put some EEG headbands on them, single channel EEG, to measure neural activity. And we saw that the people who were more engaged with the practice, people who had increased alpha and high beta brainwave activity, had better mood outcomes after the practice. But people who fell asleep during the yoga nidra, and this is one of the reasons we wanted to do the study, is sometimes so yoga nidra is pretty popular with tactical populations. So military, police, firefighters. And it's a way that they can rest their brains when they're not necessarily falling asleep. And it could be a countermeasure for fatigue. One of the concerns that I have when I see people doing yoga nidra is that they, especially if they're sleepy, they fall asleep. The problem with that is if you're falling asleep for a few minutes, it's probably not enough sleep to really give you the beneficial effects of napping. So napping in general is great, but we really want to think of, on average, like a 23-minute nap as being enough time to maybe get some cognitive restoration. So these quick little micro sleep outs that might be happening during yoga nidra or during some of these non-sleep deep rest periods, turns out they're not helpful. So drifting in and out of sleep during the practice, as measured through theta and delta activity, positively correlates with negative mood states like confusion and fatigue and anxiety. So when you're doing these non-sleep deep rest practices, may be counterintuitive, but the focus really is on non-sleep, and the more engagement that we saw with the practice, the better the mood outcomes. So it's not working like a 30-minute nap. So we also just wrapped up a second study. So we did a follow-up study with yoga nidra. And in this study, we went ahead and we had a control condition. So the same person, and this is a person who teaches iRest. She's for people nationwide. So we had her do the iRest condition, the yoga nidra condition, but then we also just had her read. So we controlled for the person's voice and the cadence, but she was just reading a book about yoga nidra. So we just finished that, and we're really excited. The data are sitting on my really good friend and colleague, Dr. Banks. So these data are sitting with him right now, and he's working on it. So he's one of my very best colleagues and collaborators. So we have a really fun relationship where we always joke around how we're always failing each other. So either I'm waiting on data from him or he's waiting on data from me. But stay tuned. Very excited to see what we get with these the second study. Another study that we just worked on this year is lucid dreaming. It may sound a little weird. So lucid dreaming, this is when you know that you're dreaming. And I know I was a little hesitant to do this study because I was worried people would think it was a little too woo-woo, out there. But then I remembered, like, back when, sort of dating myself, but back when I was in college, that's kind of how people thought. That's what they thought about meditation. That was sort of, nobody was really doing it. But then we had some people like Richie Davidson, who were, I think, one of the first people to really jump in there and study the neuroscience of meditation. There are some studies to suggest that lucid dreaming, the ability to know that you're dreaming, can help with some clinical states like PTSD. And to me, it makes intuitive sense because if you think back to that initial graph, and we have those REM sleep bouts, if we add those together, we're spending about 90 minutes at night in REM sleep, this very emotionally charged sleep where we're having these vivid dreams, our body's paralyzed, adrenaline should be shut down. And one of the main outcomes of that is processing emotion. And we take REM sleep away from people, they have mood problems, right? Increased anxiety, increased depressive symptomatology. So to me, it makes good intuitive sense that if your brain is willing to paralyze you and scare you while you're unconscious for 90 minutes a day, just to process your mood, it's probably pretty important. And so might lucid dreaming be a way to optimize that time? Your brain is processing your emotions anyway. Can we get in there and optimize that time for mood management? So with this one, we did some baseline measures, a working memory task. We looked at their sleep quality. We looked at their emotion processing, did 3 days of baseline EEG while they were at home. And then we tried to induce lucid dreaming. And this is hard to do. About 50% of people can do it. This is a senses-induced lucid dreaming technique. So this is one of the techniques where you wake up. So we would wake them up through an alarm. There's an app for this that works with the EEG headband. So we would wake them up, and this is what's considered a wake back to bed routine. So usually they'll cycle through 3 different sessions where they'll activate their senses. So we'll have them do tactile, so vibration, visual, and auditory check. And these, that's part of the SSILD feature. Some people will be able to fall back asleep if it's the second part of the night, usually going back into REM sleep pretty quickly and lucid dream. So this is a pretty big population, but 79 participants in the study. So we didn't see any association. So some of the participants were able to report that there's a lucid dreaming questionnaire. Some participants they've self-reported that they were able to lucid dream. Despite that, we didn't see any relationship with mood and lucid dreaming. So, could be one of the things that Dr. Jonathan and I talked about is, might it be that, this was just a few days. We gave them 3 chances to lucid dream. Maybe it's something that might work longer. Maybe it doesn't work at all. But what we did see is pretty interesting. And what we saw is that exactly what we would expect to see. Time spent asleep mattered, right? If you only good things happen when you sleep. So the people who had just longer sleep time had fewer mood disturbances. But it's really REM sleep. REM sleep was significantly associated, time spent in REM sleep significantly associated to better overall mood. And then if we split people into a high and low REM sleep group, we also see that within that working memory task, that reaction times were different. During this working memory task, we gave them distractors that were emotionally valenced. And we see that the people who had high REM sleep didn't really have a significant difference in how long it took them to respond to the negative or the neutral distractor. But the people who didn't get a lot of REM sleep, they were in the low REM sleep group, there was a significant difference in their response time, whether or not that affective distractor was a negatively valenced or not negatively valenced. And you might notice too that the reaction time is longer overall for, if you just visually. So one of the things that I'm wondering is, maybe increased REM sleep makes you a little bit more cautious, slower to respond when you're being presented with distractors, and some of them are emotionally valenced. So it's something to look at. So we just looked at these data yesterday, Jonathan and I, and so we're already talking about a follow-up study, and I think that's the next study I'd like to do. I think I'd really like to take a deeper dive into REM sleep and really see how, maybe not lucid dreaming, but how can we optimize that time spent in REM sleep to promote better overall emotional health. I think would be interesting and important. And also, the other part of that is slow wave sleep, because as we get older, we're not as efficient at getting slow wave sleep with age. Deep sleep, I know this, like, I don't know, I have 19-year-old twins, so my son, he sleeps, it's so beautiful when I watch him sleep. It's like the sleep of the innocent. It's the sleep of somebody not paying the mortgage. Like, he is so deeply asleep, and I can't do that anymore. Like, a wind hits my cheek and I wake up and panic. So that's kind of where we are now. And again, these are probably the two goals I have, really: increase slow wave sleep time, especially in older adults. This is very selfish because I'm getting older. My slow wave sleep isn't as good. Especially thinking about the role of slow wave sleep this is where all that lymphatic activity takes place. So especially people as they get older, and we're thinking about like neurodegenerative processes that rely on brain clearance. So maybe optimizing slow wave sleep in older adults. And then the other part of that would be REM sleep, optimize that time spent in REM sleep, especially I think in younger people would be pretty interesting. So again, I promised you we were going to see this graph again. So this is and I wanted to show you my other, Lisa Robeson insisted I put both of my dogs on here. So this is my other dog, who is her favorite dog, Pebbles. And I just want to thank my collaborator. This is Jonathan Banks, who I talk so much about. This is the great, many of you probably know the great Rob Smith, Dr. Fins, Dr. Lopez, Travis, who was part of your group, is one of my very favorite people, and my husband, who I also do research with. And these are my students. The ones in red I just want to give them a little shout out, because these are students who came through our MS in experimental psychology program, and they've all gone on to either earn PhDs or they're currently getting their PhDs. Bharath and Shravya are doing the yoga nidra stuff with me. Arjuna is working on the gut microbiome study, and Isaac did a lot of those initial sleep deprivation studies. So I'm really lucky that I have, I think, some of the best colleagues and students, which makes all this so much easier. So, thank you.